I'd like to welcome Dr. Marinka Zitnik on behalf of our Boston Protein Design and Modeling Club, and thanks to everyone for coming out tonight. We should have a really exciting conversation. Just as a little bit of background, if you're not familiar, Dr. Marinka Zitnik is an associate professor in the Department of Biomedical Informatics at Harvard Medical School, associate faculty at the Kempner Institute for the Study of Natural and Artificial Intelligence at Harvard University, associate member at the Broad Institute of MIT and Harvard, and affiliated faculty at the Harvard Data Science Initiative. Her work spans the fields of graph neural networks, artificial intelligence, machine learning interpretability, drug discovery, and protein design, all of which are very of interest to people who come to this event. So on behalf of the whole club, we welcome her, and I'll let her take it away from here.
Thank you. Hello, everyone. Excited to be here with you today and share some of our ongoing research in the group that is related to developing AI models that can empower biomedical discovery. Throughout this seminar, I will highlight several research projects. They all come with open data sets, open models, tutorials, documentation, evaluations, GitHub repositories, and so on. So if you are interested in learning more about any of those, you can go to our lab website and you'll see pointers to those projects there. Okay, so let's start. So over the last several years, we have seen an increased interest in the development, applications, use, and evaluations of AI across areas of biology and across areas of medical research. And AI is beginning to have an impact on the way we go about research and scientific enterprise into different ways. So first is various types of tools are changing the way researchers perform their everyday research activities. And second, AI models are now increasingly being used to augment scientific research process and even changing the scientific method itself, providing insights that might not have been possible with traditional deep learning models just a few years ago. An example of that is also drug discovery as an important area of scientific discovery and biomedical discovery. And what I'm showing on this slide is a highlight of a study that was published just 2 months ago that reported the first successfully completed phase 2A clinical trial of a candidate drug that targets the NIK for idiopathic pulmonary fibrosis. And the reason why this is an important early example of a success of how AI coupled together with very thoughtful chemistry and biology can streamline and accelerate drug discovery, because it's one of the earliest examples today where we can see that AI models that were used here both to nominate therapeutic target and then identify chemical compounds and optimize those molecular chemical compounds to modulate and bind to the target that was nominated, have led to an accelerated pipeline where in this study they were able to go from the initial hypothesis to a phase 2A clinical trial then in less than 30 months. And the results of the trial showed that this particular compound has favorable safety profile and shown early signs of efficacy. So it's one of the earliest examples that show that this type of AI models that we are developing and evaluating can go beyond academic benchmarks and really have an impact on drug discovery and development as one area of biomedical discovery. In my research group, we are very excited about the paradigm that is quite pervasive now in the AI field, and that is the paradigm of AI scientists. And some of you might be wondering, why are the quotes around AI scientists? And so the idea here and the vision is to develop increasingly capable and powerful AI models, models that have conversational interfaces that can serve as co-scientists or co-pilots or research assistants, collaborators to human scientists in the discovery process. These AI models integrate multimodal data. They integrate multimodal data into models that can make predictions that can be validated through experimental platforms. And the goal is that eventually these models would be able to generate interesting scientific hypotheses and operate somewhat autonomously for specific sets of tasks. So this is the broad vision that the field of AI and the interface between AI and biology and science is very excited about. So what I want to share with you today is a few vignettes that show the progress towards realizing that vision. And I will share vignettes at 4 different scales of biology that we are developing AI scientist models for. And so we'll start with the scale of molecules. We're developing models that can help us better understand the functions and the roles of proteins across a broad range of the human proteome. Then we'll go one scale up when the role and the function of proteins need to be understood in the context of molecular interactions, where we need to model not just one molecule at a time, but complexes to understand how they interact and how those interactions vary across biological contexts that can be defined by cellular environment and cell types. And finally, we will see how models that have understanding of behavior of molecules at the molecular and cellular level can help us better identify promising therapeutic opportunities and promising drugs that can be then tested in human patients. Okay, so let's start with the first part, which is that of molecules. And so the motivation for that is the broad observation that I think everyone here in the audience is familiar with, that as a field, we now have quite several models that are able to predict protein structure from protein sequence for many important areas of and classes of proteins, and that some of these models achieve atom level accuracy that matches experimental efforts. And so that has been really a tremendous grand challenge that models that predict structure from sequence have made tremendous progress towards over the last several years. There is, however, another grand challenge that remains largely open, and that is of developing models that would be at the level of generality and utility as some of those structure prediction models, but would be able to predict the role, the functions of proteins. So models that would take as input information about the sequence, the structure of proteins, and predict the functions or phenotypes of those proteins. Can we develop more general purpose or foundation style models that can predict functions of proteins very broadly? Why do we want to do that? We would want to do that because many of the human proteins are still poorly functionally understood, poorly functionally characterized. If you go to some of the canonical databases, such as UniProt, SwissProt, and others, there are thousands of human proteins that have fairly empty pages. So we really don't understand what they do. And so understanding the protein function is such a fundamental task that is relevant for a variety of downstream understanding of biology. So the statistics is that 9 proteins lack understanding about how the function of a protein changes across biological contexts, such as cell types in which protein is active. So can we develop models that can predict function for every protein that would be helpful in the same way as models that can predict structure for those proteins? So I want to share with you a model called Procyon, which is a multi-model foundation model that can do exactly that. So Procyon is a model that can model, predict, and generate protein functions and protein phenotypes. And over the next several slides, I will explain what we mean when we say multi-model and what we mean when we say foundation model. But conceptually, the way you can think about Procyon is a model that is trained once, and once it is trained, it can be used for a variety of downstream tasks. It can help with understanding of protein functions and retrieving and finding other proteins that have the function described in the input prompt. It can associate functional impact with substructural units of proteins in terms of protein domains. It can model pleiotropic effects that certain proteins might have. It can associate disease mechanisms or candidate mechanism of actions of drugs with a specific protein. So it operates across different knowledge domains. Every row here is a separate knowledge domain. So how can we think about Procyon once the model is trained? So human scientists interact with Procyon through an interactive promptable interface that is shown on this slide. And there are 3 modes of operation that Procyon supports. So in the first mode of operation, human scientists open the prompt and they can describe in plain natural language their biological process of interest. And that biological process, that text prompt, will then trigger the Procyon to go and search through its internal database of proteins and prioritize, rank, and score those that likely have the function described in the input prompt by human scientists. The second mode is a question answering session where human scientists in their input prompt can now interleave the textual description of biological functions with information about the sequence and or structure of the protein that they are interested in. And then Procyon would engage in answering some of the questions that human scientists would ask. And finally, panel C shows generative component of the model, where the model not only prioritizes, retrieves, or predicts scores for predefined for proteins in the input prompt, but instead the model generates free-form text descriptions that can be thought of as captions of proteins or protein cards and capture predicted phenotypic effects that the particular protein encoded in the input prompt would have. So you can think of panel C as an analog of what are the variety of models that you might have been using in your free time to generate captions of images of cats and dogs, but we are not here interested in generating captions of images of cats and dogs. Instead, we are interested in generating captions or cards that describe the functional effect of specific proteins. Okay, so once the model is trained, this is how human scientists can interact with them. It's a very accessible model because it's conversational in that regard. Okay, so now let's spend a couple of minutes going through the process of what is the data set necessary to train such a model? How do we train that model? And once we train it, how do we evaluate the model? Okay, so first the training data set. So now to train Procyon model, we have built a training data set. And this training data set consists of 33 million data points, where each data point can be thought of as a tuple, and that tuple consists of two elements. The first element is the protein, and it can be think of it as a protein ID that can then be linked to the sequence and the underlying structure, either predicted or experiment by your favorite structure prediction model or experimentally verified. And the second element in the tuple is a free-form text description of the role of this protein that would come from a variety of experimental data or curated databases. So there are 5 knowledge domains that those textual descriptions capture, including biological functions, disease mechanisms, mechanisms of actions of drugs, protein domain and functional impact of the domain, and molecular interactions. In addition to this instruction tuning data set that is multimodal because it links protein structures and sequences with textual descriptions, we need to have additional information that can help us train the modality-specific encoders. And so to train in the sequence models, there are like millions of sequences that the protein language model is trained on, then 800,000 of structures that include the mixture of predicted and experimentally verified structures to train the structural encoder of proteins, and 50 million full-length scientific papers that the model encounters to enable natural language understanding. And so this data set is then used to train Procyon model and train it in a thoughtful manner. What that means is that instead of pretending that amino acid sequence is just a sequence of letters and those are English letters, so it's just like a word, the same as some kind of English word in the English vocabulary, instead of making such naive assumptions, what Procyon does is takes into account that different data types or different modalities cannot be mixed in the raw inputs, but instead need to be tokenized and then presented and integrated in the input model in such a way that tokens, which are generated to represent text, are interleaved with tokens that are generated to represent sequences and structures, and all used to train a transformer-based model that requires tokenized input representation. Okay, so at the high level, once we have this training data set, we use it to train Procyon model. So what this slide shows is a high-level overview of the architecture of Procyon model. So we can read this from left to right. What we see on the left are sample prompts from the user. And so fundamentally, the human scientist would provide either a predictive prompt, so a prompt that requires some kind of retrieval, ranking, or predicting scores for a protein that is given in the input prompt, or generative prompts that require Procyon to generate text describing the protein in the input prompt. So if we read that from left to right, what happens is that the user input prompt is decomposed into elements that cover different data modalities, so that a text-based portion will be processed by a text-based LLM. A sequence-based portion of sequence in the prompt is processed by a protein language model, and a structure-based portion in the input prompt, if provided, is passed through an atom-level structural encoder. And then additional projector layers are trained in the model to align the outputs of these models so that we can generate a single unified embedding space from which we can then either do retrieval to retrieve and rank proteins, or to perform autoregressive text generation to generate in an autoregressive manner the sequence of words that would describe the function in the phenotypic description for a protein in the input prompt. So to train our model, this architecture with the data set that I just described, we employ three objectives. So the three main objectives to train this model are the following. So the first objective is the language modeling objective. The language modeling objective here is the form of text token prediction. And the purpose of that objective is to make sure that the model, when it's asked to generate text, will generate coherent scientific text. The second objective is contextual protein retrieval. So for contextual protein retrieval, we use a form of contrastive learning, which is a key objective for self-supervised learning. And it is important to have it here so that the model during training time, it can learn to retrieve and rank very highly the protein that is the one that most likely has the function described in the input prompt. And finally, the third objective is an instruction tuning objective. So this is objective that is often used with multimodal in conversational models. And the purpose of that objective is to ensure that the model will follow human instruction. So in the input prompt describes a specific biological process. Well, the model should retrieve proteins with respect to the process that was in the input prompt, not hallucinate about something else. So these three objectives were then combined into a single overall objective to train a model on the data set that I described using the neural based on the architecture that we have seen in the previous slide. And finally, that produced a single Procyon model that is a large model that has 11 billion parameters, and it's openly available to everyone to use and additionally fine-tune. Okay. So now it would be a great question or like time to ask about, great, Marinka, you spent many, oh, is there a question? Oh, go. Yeah. Yes.
Did you train everything from scratch or are there pre-trained LLMs?
That's a great question. So there are pre-trained protein language models and text-based models that we started from. So a question to ask now is that, well, what makes this model a foundation model? And so foundation model and this specific descriptor is nowadays used for models that have a number of capabilities and can perform a number of tasks, including tasks that they were not explicitly trained for and encountered during training time. And so over the next couple of slides, I want to walk you through some of the benchmarking results and then applications of Procyon to new biological problems that we have performed together with collaborators to demonstrate that capability of Procyon. But very broadly, there are four key features of Procyon that make it unique and with respect to other models that can predict protein functions. So these four features are A, multimodality. By that, what I mean is that it's possible that in the input prompt, user can seamlessly interleave textual information with information about protein sequence and protein structure. The second key capability is zero-shot task transfer. We will see that in a few applications in the following slides, but zero-shot task transfer means that the model can perform and identify proteins with candidate function without having seen any, meaning zero examples of proteins with that function during training time. And kind of models that are large and foundational in that sense are often tested for the capability of generalization to new tasks or new biological functions beyond those encountered in training time. The third capability is unbound vocabulary of user input. And so those who have ever performed gene set enrichment analysis, reactome pathway analysis, or other kinds of analysis might appreciate that because net promptable interface that Procyon has, we are not limited a priori to consider only those biological functions that correspond to pre-existing gene ontology terms or reactome pathways that are standardized canonical functions in gene ontology, reactome, in other databases. That has opened us to exciting applications where we're able to look into pleiotropic functions or functions that do not yet correspond to any given gene ontology term. And that is possible because the input is unbound in the vocabulary, right? So it's possible to just describe the function in plain English. And fourth, the model has predictive and generative components. Okay, so let's look at some of those capabilities. And what we will start with is broad benchmarking of the model. There are many, many plots like this one that we have in the paper, in the supplement. So those who are interested to see the benchmarking results where Procyon is compared to simplified variants of itself, as well as to existing models under different setups, are strongly encouraged to look at those plots that we have in the paper. So here I have just a single slide where we have tested Procyon and benchmarked it on predicting functions across different functional categories. So those are different plots. And for each plot, we have a number of models that Procyon was compared to. And so we see Procyon in the last row in purple, and other bars represent models that are emerging multi-model protein text models, such as Biotranslator and ProtST. Those are also models that integrate textual information with information about the structure or the sequence of proteins. And we can see strong lifting performance between those models and Procyon, which is in part due to the richness of the training data set and the architectural design of Procyon. Then we also include comparison of Procyon to state-of-the-art protein language models, such as ESM2 or ESM3, that can be then used in a zero-shot manner or additionally fine-tuned for modality adapters. And also structure-based models, such as GearNet, which is shown here, which is an atom-level geometric deep learning model that can be used to predict protein functions based on the structure of the proteins alone. We also include here as a comparison when the traditional bioinformatics workflows that relies on using sequence comparison algorithms such as BLAST in order to transfer functional associations from one protein to the other based on sequence similarity and evolutionary information. What we can see across in these benchmarks is a consistent boost in performance across a broad range of functional categories achieved by Procyon relative to these single modality models or more traditional approaches. What I'm showing here on the right is an example of another test that we have performed. And this was the test where we were specifically looking into some of recently characterized pleiotropic functions that would not correspond to any single gene ontology term. And we test performance of Procyon shown here in purple, and higher percentile indicates better performance. It means that the protein that has the function described in the input prompt was ranked very, very, very highly. So was very close to the top of the prioritized list when Procyon was asked to rank all of the human protein coding genes in its output. And what we can see here is that when the input prompt was a mixture that would correspond to 2 or 3 or even 4 canonical biological functions that have direct representation in standardized ontologies or vocabularies, we can see the lift in performance that cannot be achieved if one would just do prediction for each gene ontology term individually and then somehow aggregate those predictions by combining the ranks. And we have several examples of that show that the benefit of having the promptable interface towards modeling pleiotropic effects. Yes, there's some question.
Like, how can we the compensation from the training data?
Yes, that's such a great question. Perhaps let me repeat it first for the online audience as well. So the question was, what about contamination of the training data? In particular, how can we exclude and how we can be confident that those protein function annotations that the model is tested on here have not been encountered by the underlying text-based model or that was trained on scientific papers. And so that's a great, valid question. And we spent probably at least a dozen of pages in the supplementary material describing this process. Here's where we have designed a series of challenging data set splits. So for example, one was that we performed these time machine experiments where we effectively say, okay, we can go to gene ontology, we can see when are specific gene ontology terms added to the ontology, and then use those recently added annotations and stratify model performance in terms of how accurate the model is when it is asked to reason about protein functions that were added to gene ontology many years ago versus those that were added last month or 2 months ago. And you would expect from a model that was better performing model that we would see relatively stable performance of the model across a broad range of time. There is another test where we have, like, here's another question that you should ask me, which is, hey, Marinka, like, sure, the same biological pathway is actually described in multiple databases. It's not just gene ontology, it's also in reactome pathway and in OMIM and DisGenet. Great question. Because of that, we have a series of dataset splits where we have created clusters of biological processes and put all of them in the test set. So we would say cell apoptosis, all of the gene ontology terms and reactome pathways and OMIM pathways that would relate to this process would be put in the test set of the model so that the model would not encounter them during training time. Ultimately, yes.
Follow-up
Yes.
questions next comment. So
Sure.
I feel like instead of doing this like training testing separation of the same goal term, it's probably more meaningful to predict a recently created goal term like a new function, like some new cellular processes is never realized before, but you have zero-shot ability to do.
Yes. Absolutely. So I agree. That's why the benchmarking is slide number 1. And the remaining 15 slides that we will go to now are about new biological applications. And so I think there's, no matter how thoughtful, careful data splits are, and we are very thoughtful and careful and had many, many data set splits, there is no way one can guarantee to you that one specific annotation that would be predicted by the model was not somehow leaked into the training data of the underlying text LLM. So what I will show over the next several slides are now increasingly more challenging examples, including a prospective and independent experimental validation of novel predictions made by Procyon, which allow us to address the skeptic comments, especially from our experimental collaborators who would say, well, this is just doing nothing more than but being a parrot to our knowledgeable biologists. So let's start with some of those. Let's go to some of those applications. So in this, the one that I will show on this slide is an example where our collaborators were interested in identifying associations between the functional and substructural subunits of proteins, specifically domains, and how they would relate to the binding of small molecule compounds. And so here, the collaborator designed a prompt shown on the left that contains information about the function, information about the structure of the small molecule compound, miglitol in this case, and a database of proteins domains, and asked Procyon to prioritize and rank those domains in order to identify which are those that would be bound by this drug. And so the measure of success here is whether the domain that was bound by miglitol would be ranked number 1 and be assigned high score. And that indeed happened for MGAM, and there are several dozens of other multi-domain proteins that we tested the model on and asked each time whether those drug binding protein domains are domains that are ranked very highly by the model. Here's another example. So here are collaborators who are scientists studying deeply Alzheimer's disease, were very interested in starting associating the effects of mutations in the sequence of the protein with the impact that those mutations would have on the function of the protein. And so what the experiment that they designed was to take PSEN, PSEN1 and PSEN2 genes that are studied for Alzheimer's disease. And so what they have designed a series of prompts. Shown in the first one is a prompt that contains wild type sequence of the protein. And they asked Procyon to generate phenotypic descriptions of that based on that wild type sequence of the protein. And then they started introducing mutations at specific positions in that sequence and testing how predicted functions for change when the input to Procyon is that mutated sequence of the protein. And then looking at the functions that were predicted with the mutated sequence and are not predicted now, were predicted with wild type sequence and not with mutated sequence, allow us to start associating those differential functional impacts with positions on the sequence and then overlay that on the structural information. Here is a third example. And so here our collaborators were really interested in understanding the functions of proteins that are poorly functionally characterized. So one of these proteins was AKNAD1. AKNAD1 was recently implicated in Parkinson's disease. Because of that, would be great interest to understand what does AKNAD1 do. When we look at canonical databases for AKNAD1, the functional pages are relatively empty and the annotation score is low. When we would look at sequence similarity between the AKNAD1 and other proteins that have known functional annotations, sequence similarity is relatively low, meaning that an approach where we would be able to transfer functional annotations from other proteins to AKNAD1 would not be particularly successful. Okay, so what we have done here is, I will illustrate that for AKNAD1, and this process can be repeated for many proteins that have empty functional profiles. So here we're going from left to right. In the input prompt, we provide information about AKNAD1, and then Procyon is being used in generative manner to generate now phenotypic descriptions. One of the descriptions generated for AKNAD1 was about putative involvement of the protein in the formation of the actin cytoskeleton. And now to make sense out of that prediction, we have looked into the human protein atlas and specifically information about subcellular localization of this protein to find an evidence that can support this prediction generated by the model. And so this process was now repeated for a few different descriptions for AKNAD1 and as well as various other proteins together with our collaborators. I will show two more applications that are, I think, quite exciting. So this was joint collaboration with Pentuliti Lab from the chemistry department at MIT. And they were very interested in using Procyon to predict peptides that would likely bind ACE2. And so in order to support this application, here's what we have done. So we took Procyon pre-trained model, and then we fine-tuned Procyon model using a small data set of naturally occurring protein peptide binders. So this process of fine-tuning gave us a version of the model that we refer to as Procyon bind. And so now we use Procyon bind model to prioritize candidate peptide sequences. And so then our collaborators were able to synthesize those peptides and perform binding assays to identify whether those highly ranked peptide sequences were indeed stronger binders for ACE2. And what we can see here in panel 4 is how the correlation between the predicted score by Procyon bind and the actual then experimental measurement was such that those with higher binding score were ranked and retrieved highly by Procyon model. Most recently, we worked with collaborators from Imperial College London and King's College London, and so they were interested in characterizing genes that are involved in pathology of multiple sclerosis. And so what neuropathologists at UK have done is they designed a series of prompts, specifically 5 prompts, some of which, the beginning of which we can see on the left part of the slide. And each prompt describes a specific pathology that is seen in patients diagnosed with multiple sclerosis. And for each of those prompts, the task to Procyon was to identify, nominate proteins that are involved and can explain that pathology. And so to validate those predicted lists of proteins, then our collaborators collected 280 post-mortem brain samples, stained them, and perform RNA-seq analysis to identify genes that characterize each of those states of each of those pathologies, and compare those experimentally determined lists with the list of proteins nominated by Procyon. And these box plots that we can see in the middle of the slide show where in the ranked list of Procyon, so higher percentile is better, were located those proteins that were found through this independent experiment that they performed. And we have seen across all of the 5 different prompts. They describe different pathologies of MS. The relevant proteins were retrieved very highly, and then collaborators use those descriptions generated by Procyon for those proteins to further mechanistically understand the role of proteins in these specific areas of MS. So these are just a few vignettes, and there are several more that we have performed with different collaborators to demonstrate the broader utility of the model, where the model can be used to predict and characterize biological functions that did not encounter at training time because were de novo specified for the first time by collaborators and evaluated in a perspective against experimental or independently generated data. There are many more, and this kind of broader use of the model establishes the foundation for the model to be broadly applicable to tasks that relate to understanding of phenotypes and functions of proteins. So this is about Procyon. You can see how under the AI scientist paradigm, Procyon represents a foundation model that can help us provide functional insights about the roles of proteins. But of course, proteins do not act independently of each other, but instead they form molecular interactions and even broader molecular interaction networks, and that are often responsible for executing and activating important molecular pathways. So what I want to share with you next is the work that was recently completed by a stellar PhD student in the group, Ada Feng. And Ada was very interested in the question of molecular interactions and how to represent molecular interactions better in a way that would allow us to model sets or complexes of molecules, not just one molecule at a time. And so she developed a model, Atomica, which is a representation learning model, meaning that it's a model that generates latent representations or embeddings of molecular interactions, specifically interaction interfaces between molecules. So what does that mean? That means that for a given molecular complex, we can extract specific interaction interface, and Atomica will generate an embedding or latent representation or signature of that interface, and then be able to do that for many different types of molecular modalities. Algorithmically, Atomica is an atom-scale geometric deep learning model that is trained on graph-based representation of data. So unlike Procyon, where the data was multimodal and consisted of predicted or derived structure sequences and text, Atomica is trained on graph-based data set that represents interaction interface. So the way we build this graph-based data set is that we start with interaction complex, and one defines an area of the interaction interface and then represents it with a graph. And in this graph, we have nodes of 2 different types. We have nodes that represents atoms in molecule 1, nodes that represents atoms in molecule 2. Edges between those atoms indicate information about bonds and additional attributes in terms of bond length, dihedral angle, etc. And then the second level of information is information that encodes how those atom level representation maps to higher order structures such as common chemical motifs, amino acids, or nucleotides. That now means that for each and every interaction interface, we have this bi-level graph representation. And we have now millions of those bi-level graph representations. So that gives us a data set of 2 million here the bi-level graph representations of interaction complexes. And on this data set, we train an Atomica model. So Atomica model is a representation learning model, as I mentioned. So the goal is to identify signatures or embeddings of interaction interfaces. And it is trained in a self-supervised manner, meaning that it does not require additional information about the labels or the functions of those interactions. And specifically, the pre-training objective of Atomica is to take a particular noise interaction complex and mask specific blocks in those complex. And Atomica is asked to predict the identity of those masked blocks. So you can think of that as a one-step diffusion process that is defined that Atomica is tasked to solve during the process of training. Now, the advantage of Atomica being an atom model that operates at the level molecules. Instead, we can train a single model across different types of molecular modalities. And in particular here, we train Atomica model to the self-supervised denoising and masking objective on 2 million interaction complexes that include complexes involving small molecules, proteins, lipids, ions, and nucleic acids. The output of that training process is now the latent embedding space, which you can see here in this right part of the slide, where every point, and we would have 2 million points in this latent embedding space, represents an interface between a pair of molecules. And so is this latent space now useful? Right? So how can we test this in a way that would demonstrate both the utility of this latent space as a resource and also help us provide new biological insights? So again, we started with a number of sanity checks where we expect or know about, have specific knowledge about the chemical structure, the processes or the functions of molecules, which can serve as a way to test whether the organization of the latent space of Atomica aligns with the expected behavior. So for example, we can look at embeddings of interfaces, which we can then probe further to get the embeddings of individual atoms or embeddings of amino acids and nucleotides and see whether those embeddings cluster in a meaningful way by types of atoms or types of amino acids. Fairly simple test, but worthwhile always doing, and we see positive results there. We can also look, yes, questions.
Existing cluster with the DNA.
Yes, so that in this previous slide, right? Yes, so that's a great question. It was because of the oversampling of certain structures that were less common in the training set. So it's an artifact that we try to explain through then identifying what are the weights that the model assigns to individual atoms when learning representations of the molecule, which we do through this score we call Atomica score that allow us to decompose an embedding into the weights that are assigned to individual atoms in the graph level representation of the interaction interface, and that can then identify potential biases in terms of overrepresentations of molecules of certain types that would suggest to the model that the importance or similarity between certain atomic units is greater from biological relevance standpoint, but it's only greater in the data set the model was trained on. So it's a number of these type of investigational biases where the data that are used that are generated to train these models are not uniformly sampled from the underlying hypothesis space, which can lead to the biases that would then influence the organization of the embedding space, which we can probe with Atomica score and then use to additionally reweight during model training to accommodate and account for those biases as much as we can. I think there was another question there.
For the
yes.
Slide, did you ever analyze do the same analysis but at the level of functional group?
Yes, so that's a great question as well. So we have also looked at a number of functional groups, including protein domain families for proteins. There are for other molecular modalities. There are also some somewhat smaller incomplete data sets that we have used to perform functional enrichment tests as a way of probing the latent embedding space.
At what level would that be the most structured one?
Sorry?
So at which level would they be the most structured?
So the strongest structure is at the level of interaction interface, where one embedding is generated not at the atom level, not at the graph or block level, but the level of the entire graph representing interaction interface, which is not surprising because the model is trained to primarily optimize for the organization in the embedding space at the level of interaction interfaces, meaning that two interaction interfaces with the similar structural properties would be embedded close together. That is the kind of the primary objective here, rather than learning atoms or common chemical motif level embeddings, for which there are already quite a number of strong models out there. Okay, so one of the key motivations for Atomica in this development was to see whether we get any additional benefit from representing data modalities that have relatively limited training data by training a single model across multiple data types. And so what we are seeing here is we performed, well, let me first explain the experiment where we performed. We trained a set of Atomica models. So we trained one Atomica model on all molecular data modalities that were in the data set, including small molecule interactions, protein interactions, ions, lipids, and nucleic acids. And then also trained reduced, simplified versions of Atomica models that were only trained on one modality at a time. And so then what we wanted to understand is whether that larger model that was trained across different modalities is able to generate better representations than models that were specifically tailored and trained in a specialist manner for each data for data modalities separately. And so what we will see here in this next slide on the left, where we see the bar plot, we can see performance of a joint Atomica model that is trained on all molecular data modalities, and then compared to performance for this specific task on this slide. I'm showing the ability of the models to identify the identity of the block that was passed from the molecular interface by reduced versions of Atomica models. And so what we can see is that across all molecular modalities, there is a benefit to having a single model being trained on the query modality as well as others, and that boosts performance and leads to better generalization. In particularly, if we now generate the plot shown on the right part of the slide that shows on the y-axis the change in AUPRC score, which is what is the improvement or performance gain from having the model be trained on multiple data modalities. And then on the y-axis, we see the number of molecules of a specific data modality in the training data set. What is exciting here to see is that the lift in performance is the greatest for those molecules, molecular modalities such as protein DNA interactions and protein RNA interactions, for which the data are most scarce in the training set. And so the reason why this is interesting from the perspective of AI for molecular sciences is because we are kind of interested when we train very large models. If training a large model can provide benefits that cannot easily be explained from training a smaller model. In order to get a sense whether training larger models is useful and goes beyond just an exercise of being able to scale up an engineering of a model to a larger number of GPUs. And here it's exciting that we are seeing particularly the lift in performance is the greatest for modalities where the currently training data is most limited. Okay, so there are a number of other analyses that we have done to probe the latent embedding spaces or fine-tune embeddings towards downstream tasks that relate to predicting binding affinity and binding scores and others that I will skip in the interest of time. I will just show here an example of analysis that we have recently performed where we wanted to use Atomica to generate new hypotheses that can be experimentally tested. So specifically here, we used Atomica for the purpose of identifying and nominating potential candidate heme binders. And so what that meant that we started with the latent embedding space generated by Atomica. And then we fine-tuned that Atomica model to create what we call Atomica ligand. And then that model has nominated proteins that the Atomica score was very high, meaning the model was confident that those are heme binders. And we wanted to test whether we can experimentally confirm whether those proteins that are otherwise proteins in the dark proteome, meaning that they have very limited functional understanding, would indeed be heme binders. So our collaborators were able to synthesize those proteins and then perform functional assays of heme binding. And what we found is that for 4 out of 5 predictions that were tested here we were able to provide strong evidence of heme binding for those proteins. What I'm personally most excited about is that for 4 out of 5 of those proteins would not be predicted as heme binders if one would use standard structural motifs as heuristics to identify heme binders, which is kind of a typical approach nowadays, where one would not have embedding signatures of molecular interaction interfaces, but would instead scan the interaction interface and look for structural motifs that are known signatures of heme binding. And so what we show here is that it is possible to go beyond that and provide value that is complementary to established approaches. Okay, so in the remaining 10 minutes or so that I have, we will go one scale up and we'll go all the way to patients and therapeutic development and see how these models that we have discussed in the seminar so far, in particular Procyon, Atomica, and models like Procyon, Atomica, can help with therapeutic development and reasoning about the drugs in order to help patients with significant clinical need. So I hope this will be the fun part, given that it's quite late here in Boston. Okay, so what are we doing in this space? So in this area, we are very interested and are developing the AI scientists to the paradigm of agents, which are AI models that can now perform certain activities somewhat autonomously and do not require human intervention to perform certain analysis. So it's not this type of agents you see on this slide. Instead, we're thinking about AI scientists, AI agents that can help generate scientific hypotheses and help operate some of machine learning models, such as Procyon and Atomica, that we have just seen in the first part of the slide. Okay, so first, how hard is it to build those type of AI scientists? Can you yourself build an AI scientist, AI co-pilot, research assistant for yourself? The answer to that is yes. So what we have released earlier this week is Tool Universe, which is available through aiscientist.tools website. And so if you go to aiscientist.tools, you will get information about how you can now connect any large language model or large reasoning model with a scientific toolkit that consists of tools that you use in your research, which can be machine learning models, software packages, bioinformatics workflows, structure prediction models from AlphaFold to Boltz to Chai and many others, knowledge databases, and then build your own research assistants to help you with your everyday research activities. So there are 3 steps to build an AI scientist using Tool Universe. Step 1, you select a large language model, reasoning model, a general purpose AI agent, or a biomedical AI agent. This can be open source models such as DeepSeek, Llama, QN, closed source models such as Claude or ChatGPT and others. Second, you connect that model using Tool Universe to tools in Tool Universe. So you might say, well, if your everyday research involves in working with bioinformatics workflows for omics data analysis, there are a suite of tools out of 600 tools in Tool Universe that contain tools for single-cell transcriptional data analysis, differential expression analysis, cell type nomination, cell type labeling, etc. In your everyday research activities, you're using models that can predict structures and model structures such as AlphaFold or Boltz or Procyon or many other fantastic models. And those models are tools that agents will then use and execute complex workflows for you. So this is the second part, which is select the language model of your choice, connect it with Tool Universe, and part 3, your AI scientist copilot is ready for use. So if you go to aiscientist.tools, you will currently see over 600 tools that we provide in Tool Universe, as well as instructions how you can then connect Claude, Gemini, QN, GPT Codex, ChatGPT API with any subset of tools in Tool Universe. So the way this looks like is, if you are used to working with Claude, and this is how generally the website interface would look for Claude, then there is an option to connect Claude with Tool Universe. And what practically that means that when you ask then Claude about the structure of a protein, it will not just hallucinate and come up with something based on what is described in PDF manuscripts of papers, but instead it will realize that there is a model called Procyon, or there is a model called Boltz, or there is a model called AlphaFold, and it will go and execute that model and get the output of that model and present that output to you. So that allows us to realize and benefit not only from the vast scientific literature knowledge, but really connecting scientific literature knowledge, natural language understanding with previously vetted or trusted scientific tools. How is it possible? So the key insight and the key ingredient of AI agents is the ability to connect large language models with other tools, which fundamentally requires to specify a protocol that would allow an agent to decide to parse the user prompt and decide which tool to execute from the toolbox that it has currently that's currently available to the agent, then go and execute and invoke that tool, get the output of that tool, and return the results to the large language model to show and display to the user. And so frontier of AI research in AI communication has been concerned with developing this type of protocols that specify the nature of exchange of information between AI agents and scientific tools. That allows us now to think about completely new layer of infrastructure in scientific research that can be potentially comparable to the kind of applications, opportunities that HTTP protocol many decades ago has created for web. Before HTTP protocols, what existed there were many thousands of isolated web pages, and really nobody knew how to link web pages together. And with HTTPC, we were able to start adding links from one web page to the other, which then created to this type of network internet effects. So it's now possible to not only work with an isolated number of many dozens and hundreds of different kinds of models that are being developed for a specific data type or specific task that you're interested in, but also benefit from all of those models and in a productive manner. So I want to illustrate one concrete example of an agent that is powered by Tool Universe. So this is an agent that has been developed by Shanhua Gao, Zhenglong Kong, and several other students in the group. It's an agent that is called TX agent, which stands for therapeutics agent. And it's an agent that is focusing specifically on medical research. It's an agent that can reason about drugs and the use of drugs in different patient populations in order to nominate those drugs that would be safe and effective for patients. So you can think about TX agent as an AI scientist for medicine, for medical research. Once we create TX agent and connect it to Tool Universe, it then allows the users of TX agent to, in their input prompt, contain information, describe information about specific patient, about specific disease that patient has, and the candidate drug that one would consider giving to the patient or consider exploring or investigating, studying in clinical setting for that disease and that patient. And then the model will reason by using a number of tools it generated about the patient context, disease and drug context in order to answer the questions that's being asked, produce a justification to the answer that it generated, and generate a reasoning trace, which is a sequence of actions the agent performed to arrive at the conclusion that it gives to the user. There are three key features of TX agent that make it stand apart from large language models. A, it's open-ended. So again, open conversation that allows the agent to naturally converse with human scientists. Second, the model is able to perform multi-step reasoning, meaning that it can solve more complex tasks by using a sequence of tools rather than just one tool at a time. And we show favorable performance of this model against state-of-the-art large reasoning models such as DeepSeek R1. Okay, so for TX agent, we use a subset of Tool Universe, a subset of 211 tools. Those are tools that contain information about descriptions of the disease from human phenotype ontology. We have the entire open FDA made available to TX agent. So when it's asked a question about a drug, it will retrieve in real time the latest information about the drug from FDA prescribing cards, from regulatory documents, to ensure that the reasoning is grounded in trusted, verified resource, and information from open targets that contains insights about the molecular and genetic knowledge about disease and ongoing compounds in the development. The challenge for TX agent is to train this agent, given that it's trained to use tools and it's trained to perform a multi-step reasoning. And so to train TX agent, we have compiled a data set that describes each and every data set, each and every tool that is available to TX agent, generate a set of 85,000 of therapeutic questions that are questions about therapeutic reasoning, and then reasoning traces that show what tool to use in what specific tasks so that the model can understand when it's useful to call a specific tool. Okay, so very briefly, a benchmarking of the model, as you usually do, and second, two new applications, which are very exciting that I want to share with you, and then I'll wrap up for today. So first is benchmarking of the model. How we can benchmark such a model to get the sense that does this produce any useful and potential new insight beyond what it has now seen during training time, and all the questions that were asked regarding Procyon apply here, plus more, because now we have this agent model that is not just a single large language model anymore, but it's a large language model that can also retrieve data from external APIs and databases in real time. So we performed a number of experiments. One is the one that's shown here, which is that we took FDA drug prescribing cards for new drug approvals. So these are these PDF documents that were 20 to 40 pages long, that upon a new approval would be compiled by FDA, that consists of many sections. Each section can be from paragraphs to pages long and contain information about toxicology of the drug, or indications of this drug, or adverse event profile of the drug, or the use of the drug in specific patient populations, or whether the drug is contraindicated for diseases that have a specific for patients that have a specific disease, and so on and so on. There are many sections in those prescribing cards. And so what we wanted to test is whether this TX agent model can generate entire sections of those FDA prescribing cards for new drug approvals, right? And every month there are a handful of new drug approvals. We can go to those and perform this type of tests that I'm showing here, where we have compared TX agent, here an 8 billion version of the model, to other models, including models that are large language models that do not use any tools at all, models that are larger in size and do not use tools, and also comparable models that are state-of-the-art models in machine learning that also use tools similarly as TX agent. And so what we can see here, where on the y-axis, higher number indicates greater performance, stronger performance of the model, is across different sections of drug prescribing cards. TX agent performs much, much better than what is possible to achieve with these existing models. Very practically, one can start asking these questions. Just an example shown on the left part of the slide, where here the question is about a 60-year-old female patient with some history of myocardial infarction and hypertension. This person is being evaluated for medication adjustment. She's currently taking a beta blocker. She has no known renal or hepatic impairments. So given this background about the patient, is there another medication that would be most appropriate to add to her regimen? And the goal is to further reduce the cardiovascular risk for this specific patient, right? So this is an illustration of the kinds of questions that the model is asked to solve. And you can imagine that this complex question requires to, well, get information about beta blockers and their known safety profiles. It requires retrieval of information about the specific patient population, which are older female patients, and whether there are any increased risk for this patient population for the development of cardiovascular risk. And that is the process that agent engage in during the reasoning to retrieve data via its tool use from open targets, from open FDA, collect that data, and then synthesize and reason about it. So in one experiment that we have performed together with NVIDIA, we were interested in comparing TX agent against state-of-the-art reasoning models. And so here I'm showing comparison against DeepSeek model. And so for those who might not heard of it or be aware of it, this is the model that has been released a few months ago. When it was released, it really was a major shock to the current frontier AI models, because it was a model that was released in an open source manner and performed at that time state-of-the-art models that were close to commercial LLMs. And so we wanted to understand, well, how does TX agent compares to DeepSeek R1? And so here, what we see on the left part of the slide are performance of TX agent on the treatment prescribing card benchmark under the open-ended regime, when the model is asked to generate free-form text description that will be contain an answer to the question, or multi-choice regime where the model has to select whether option A, B, and C, or D, which of those small number of predefined options is correct answer to the question, right? And so we can see here the lift in performance in TX agent over 3 variants of DeepSeek R1 models, including 870 and 671 billion model. So a good question to ask now would be, well, great to show these bar plots, how can we kind of get understanding how can we explain this lift in performance? Is this really better performance, or is it some kind of shortcut that the model has learned so that it can outperform this seemingly much an order of magnitude larger model? And so what we have found is that the key capability of these models is tool use. So during the process of reasoning, these large reasoning models, what sometimes encounter is that they make a mistake early on, perhaps in step 2, and those mistakes and errors compound over time. So if one makes a mistake in step 2, then what you would do in step 3 is already correct. In step 4, which will be even worse, and step 5 will be even worse, etc. And it's hard for some of those models to then recover from the early mistakes when they have to reason over longer horizons. In contrast, for TX agent, we require each step to be grounded in trusted verified resource. So whatever the task will be done in a given reasoning step must be supported by open targets or open FDA or human phenotype ontology, or the tools that the users defined are trusted verified tools for medical research. And that allows the model to hallucinate much less than some of these alternative models. Yes, question.
Are you giving
Yes. So yes, well, DeepSeek here had access to the internet, and it was also a much larger model than TX agent. So here TX agent that was compared was an 8 billion model, whereas DeepSeek was here an order of magnitude larger. So to summarize, takeaway lessons for those who are interested in developing AI agents and this type of models, there are 2 critical capabilities that are helpful for those models to have. The first one is reasoning capability, meaning that for tasks and questions that are increasingly complex, that are not just yes or no simple questions, then the way to answer and complete those tasks is to perform multiple step inference. And so being able to go through multiple steps of inference generally leads to improvement in the quality of the final result. We are seeing that here, for example, in panel G. And second is tool use. Models that have the ability to be able to use tools have then the effects of either being better performing model, or you might have a model that has the same performance, but it's an order of magnitude smaller in its size, which makes the deployment much easier, or have models that can be grounded in hallucinate less than models that would only be able to use internet and not use any of the more trusted resources that are standard in a given community. Okay, so I will wrap here just with evaluation of novel hypotheses, right? So again, we have seen with FDA drug prescribing card generations and other benchmarking tests, analysis that give us insight into how the models are able to perform on the retrospectively corrected generated data or recent approvals. But then the question becomes, well, can the model be used to generate new hypotheses, and how can we go about validating those new hypotheses? So on that front, we are pursuing 2 directions. The first direction is that we are emulating a number of clinical trials for predicted drug candidates. So currently working with our collaborators at Harvard Medical School, Clalit Innovation in Israel, Gates Foundation, and 90 foundations in the Rarest One program at Chan Zuckerberg Initiative to simulate clinical trials for those drugs candidates that TX agent predicted as being potentially safe and effective for specific patient population. Here's just one example of a trial that was recently emulated by Ayush Nori to test the predicted protective effect of a drug called dapagliflozin, which was initially developed for diabetes for Alzheimer's disease. And so here the process entailed creating a cohort of control and treatment patients. Those were patients that were not on the beginning diagnosed with Alzheimer's. They were matched in ages, matched in sex, and other important features. And then the control group was not put on dapagliflozin. Treatment group was put on dapagliflozin for the primary indication, which is the diabetes. And we can track that population of patients over time. On the x-axis, you see 8 years. And on the y-axis, we can measure the probability of dementia-free survival. And this is one endpoint, and there are a few others, several others one can define in the context of Alzheimer's disease. And so given that we can see here the green line being above the red line, this suggests the protective effect of dapagliflozin for Alzheimer's. So we can perform now a number of those type of tests, which are now tests on real patient data for real patients on electronic health records that are locked and hidden in their healthcare systems that none of the models have encountered in any way, and allow us to test those predictions in the effect they would have for real patients. And the second type of evaluations that we are doing is that we are working with our collaborators who can generate brain organoids from samples collected from patients diagnosed with bipolar disorder. And so Ayush has here worked with our collaborators to test some of new predictions for a drug candidate that the model developed by Ayush called Cypher was predicted as potentially being effective for bipolar. And so collaborators have grown those the organoids from samples collected from bipolar patients in the clinic at Mass General, and then administered to those organoids the drug predicted by the model. And so then what we do next is we look at the response of those brain organoids to the treatment that was given to them. So in this heat map that is shown on the right part of the slide, every column in that heat map is one organ model. And so this particular heat map represents the transcriptional profile of key genes in that organ model. And so what we can see in the green group called BD treated, those are organoids that were treated with the drug predicted by the model. And you can see that the transcriptional profile here resembles the profile of control organoids, which are grown based from healthy individuals, whereas the BD untreated group is a group that has a very different transcriptional profile. And there are several other multidimensional omics characterizations that were performed here to control for various effects that could be potential confounders. So I'll wrap up here by just sharing a couple of links. So first is that if we are running an international competition at NeurIPS called CureBench. If you go to curebench.ai, you can participate in that competition or at least see what are currently best performing AI models or AI agents for clinical drug development. You can also make submissions to our live leaderboards, and we'll give out awards and start both in terms of prizes as well as travel awards to NeurIPS in San Diego in December. Second, we have a number of open resources that come in the form of data sets, models, benchmarks, and evaluations that are all available through our publications and linked to GitHub repositories with examples and documentation of use. And with that, I'll wrap up here, acknowledge some fantastic groups of PhD students and postdocs that I'm able to work with in my group, and very happy to take any further questions. Thank you. Yes.
New tools get added to Tool.
That's a great question. So the question was, how do we add new tools to Tool Universe? So adding a new tool involves specifying one JSON object. So what you need to add a new tool, what we need to provide is description of the tool. And this is generally a few sentences of text. And often we get this description of the tool from documentation of the tool. Second, we need to have then a mapping of the inputs of to the tool and the agent. And that is specified by an API, where in the JSON object we specify what are all inputs to the tool, so that the agent knows how it needs to compose a query to call the tool. Like the input can be just primary amino acid sequence, or it can be the primary amino acid sequence plus the confidence level that we want the resulting output to be optimized for. And the third input, the third descriptor of the tool involves interpreting the output of the tool, so that we have a mapping between the output that the tool provides, whether that's a score, a text, a structure, a sequence, and the way that output would be interpreted by an agent. So all this can be provided to Tool Universe through a very simple online form that we have in aiscientist.tools website, so that one can very easily add new tools to Tool Universe. Yes, question there.
So for Tool Universe, I assume the tool execution is hosted by the developer, right? So that cannot easily scale up. A lot of users all over the world. So what would you foresee a sustainable future for these types of agents?
Yeah, that's a great question. So the question was, how can we implement or deploy tools that are tools that go beyond retrieval, such as tools that involve executing an algorithm or running an inference of a model that go beyond retrieving information from a website or from a curated database. And so that's a great question. So the way we can handle those tools is by either, and what we do is to provide instructions of how one can set them up locally, or alternatively, there are tools that are now increasingly available through API calls. And so you can still use those tools by simply providing your API key that you have to access AlphaFold 3 or some other model. And then the agent would use your credentials on your behalf when it's executing the tasks, and can even be set up in such a way that before it would go and use the API that you give access that you grant, it would check with human scientists whether it's allowed to do so, and whether you agree of sending that queries. Yes, question.
I was wondering, how does the agent knowledge that AI scientists are developing compared to some tasks that already have very established pipelines? Like, is agent choosing the optimal tool for a subtask, or it's building one feasible that's it, or
Yes, so that's a great question as well. So Tool Universe has several functionalities that I was not able to discuss in detail, but one of them is tool composition. So where the Tool Universe can decide to chain tools together such that the output of one tool goes to the input to the next tool. And it can create this type of workflows itself, or the user can specify a previously validated, published, or established protocol or a bioinformatics workflow that would then consist of a series of tools that are chained or piped that the agent would execute, as long as it can correctly interpret the description of the tool, which uses to decide which tool to use from its tool to execute from its tool universe, and correctly interpret the input and output of the tool. Yes.
Yeah. Thanks very much. Usually when interacting with this language model, the system message and the prompt is very important. Giving a lot of data. Have you optimized any kind of which system message or which prompt is best for which tool, or no, the user should do it?
That's a great question as well. So the question was about oftentimes for these conversational models or large language models, the design of the input prompt specified by the user plays an important role in how useful or accurate the agent then is. So yes, that's absolutely true. So first, one of the features in Tool Universe, in addition to Tool Composer, is also Tool Optimizer. And so what Tool Optimizer does, it takes that initial description of the tool that the user provided, and then it will optimize it through advanced prompt engineering techniques to arrive at the prompt that would maximize correct use of the tool. And for correct use of the tool, we have a number of unit tests to ensure that the tool that is being used that being called and the way its outputs are being interpreted are correct based on kind of sets checks for each tool. Another comment that I'd like to make is kind of a broader, something we realized during the process, during the time of developing the Procyon model from the first part of the talk. So once we trained the model and started performing various evaluations and applications with our collaborators, what became clear is that even if we would study this, ask the model about the same biological function, if the user was a first-year PhD student, they would describe that biological function very differently than a senior PhD student, than a postdoc, and very differently from a senior PI who might have a lab for many years studying a particular biological pathway deeply and carefully. And so that led to additional insights in terms of paraphrasing phenotypic descriptions of proteins in order to accommodate diverse levels of backgrounds of scientific users and also specialties or areas of the research. We also found that cell biologists would talk about the biological pathway differently than a geneticist, than a systems biologist, than a molecular biologist, even though perhaps the biological pathway would be the same or closely related, but it would be different language that would be used, or the focus or emphasis on specific elements or roles of the molecules would be different. This is in large part completely ignored in existing large language models, but the idea of transferring style from one user persona to another user persona, where those personas are members of our scientific research communities that have different levels of backgrounds, expertise, or areas of specialization is largely open. So what we have done in Procyon is we designed a series of data augmentation strategies that allow us to take all of those instructions in the training data set and paraphrase them as if they would be described as when the user would be a junior scientist versus a senior scientist or a scientist with a specific background. Thank you. Perhaps we go there.
Predictive facts and figures being used to inform which patients get on the trial or the
Yeah, so this question was about TX agent and whether we can also use it to predict patient populations that would be at the higher risk of developing a specific adverse event, which could potentially be used for clinical trial design and optimization. So that's a fantastic question, and that's exactly one of our current major ongoing uses of TX agent to identify patient populations that are at high risk for a specific adverse event. And then that allows us to provide a more tailored precision medicine treatment recommendations, where it's not everyone with a specific diagnostic code would be recommended for a specific drug, but it would be, prediction would be made by considering other background, medication history, genetic information of the patient background. And in a separate study, we are working with clinical trial designers to use this model for patient selection or patient enrichment, which is the type of task where the question becomes to optimize the eligibility criteria in design of the clinical trial to see whether one can maximally enrich participants in a clinical trial for those that would likely be responders to a drug. And we're doing that in the context of immunotherapies. Okay. Sure, go ahead.
About this TX agent, I think it was only with 8 billion parameters.
Yes.
I assume that's just like one of the 8 billion parameters that you fine-tune, and it's doing a lot from those information that's taken from the PA, whatever. It's doing a lot, and I was just wondering if those other models that you showed have been fine-tuned also in the same way, or no, you just use what was the white box and compare.
So that's a great question. So there are a few different versions of TX agents of different sizes that we have available. Then when in our benchmarking, we have compared both to off-the-shelf models as well as models that were additionally tuned on the same data and trained on the same data the TX agent was trained on. And the third comment is that a more standard approach or established approach to developing this type of agents would be to develop them through RAG, as you mentioned, through retrieval augmented generation, which is this idea that a large language model, during the process of answering a question that is being asked to answer, it would retrieve information from some external knowledge base, just a knowledge graph or some store of embeddings. So this is not what is the case here. The key idea of agents is not to retrieve information from external database, but to use tools. The simplest form of a tool is retrieval, but retrieval is just one specific type of a tool. So there are many other tools, like an ML model, that the model would write Python code that would then execute that ML model on a specific input. And so the tool the principle is broader in its scope and applicability than a retrieval augmented generation, which was the approach that was popular before tool use capability was developed. Okay, I guess we will be wrapping up. It's already quite late.
Just because we're 30 minutes over the time we typically end, we're gonna wrap up. Happy to continue the conversation sort of offline. I didn't see any Zoom questions, but I'm sure Dr. Zitnik is available via email, and for everyone here, we'll take things offline. But thank you all for coming and for your attention.
Yeah.